Hi everyone, and thanks again, Arena, for the invitation to share our work. Um, as she mentioned, I'm in Uppsala, in the group of Philippe Maya, and my background is mostly in optical spectroscopy, time resolved spectroscopy, applied to biological systems. And more recently, I moved in sort of into the X-ray world, um, and focusing mostly on sample delivery methods for solution-based X-ray techniques at FELs. And today I just wanted to share some of our recent work regarding some transient solution scattering work we did using a high repetition rate XFEL, particularly at the European XFEL. Click there. Okay, so obviously this audience doesn't need much of an introduction to the world of solution X-ray scattering, but just as a brief overview, one can use so-called small and wide-angle X-ray scattering, SAX-WAX, to probe biomolecular structure. And generally, this is achieved by shining an X-ray beam at a sample and following the pattern of the scattered X-rays onto a two-dimensional detector. And by plotting this scattering intensity as a function of angle, as you see here for hemoglobin, you get sort of a broad featureless curve, which can be used to determine features such as the size of the protein or the shape, and potentially its oligomeric state. But it's not so useful. These are generally low resolution, not so useful for determining fine structure or dynamics um, within a biomolecule. More recently, sort of time resolved variants of this SAX-WAX technique have been developed, which enables one to resolve different scattering curves in response to some sort of trigger, such as an excitation pulse, pulse of light, change in temperature, or change in pH. And most importantly, thing to sort of grasp about solution scattering and its benefit for biological systems is you sort of retain near native sample conditions. You can study systems in their native buffer, as opposed to techniques such as crystallography, where the samples are confined into a sort of a rigid structure, or cryo-EM, where you rely on freezing the sample, which has its own potential obstacles. So just as a sort of example of what this data looks like, in terms of the transient wax, that is. Here you see some data that was collected, I believe, at Biocar's some years ago now already, by the group of Sebastian Wessenhoff. And here we're looking at phytochrome, bacterial phytochrome, which is shown in the left here. And in the three panels, you see sort of the full length structure. So in various truncations of the structure, where you start restricting down to the local chromophore domain, which is this pass domain. And you see in the full length structure, large structural changes, or a large response happening in many milliseconds, which is carried over into the Pascal phi domain, or truncation. And then when we look at the smallest domain, you see very small changes, which are very difficult to resolve, although clearly occurring, because you see in the long time traces that there are some features. So this is limited by the noise level of the experiment in this case, which is typically on the scale of 10 to the minus 4. So if you do kinetic modeling, you can apply this to the data and recover several intermediate states that might be present within the photocycle. You can also do structural modeling to try and figure out what these features represent in terms of the dynamics of the protein. But generally, this is very difficult, just because limited by the small size of the signal and the difficulties with the experiment itself. So one generally needs to, in order to boost the signal, to get the most information out of the experiment, one needs to work with fairly high sample concentrations or excitation intensities, which can hinder, obviously, or perturb the system itself. You also have limited temporal sampling. These are typically collected in sort of a pump probe configuration where you collect one time delay at a time and then build up a whole a set of delays at different times or as a function of time, and generally limited to large structural changes. So our question is, is there a way that this can be improved? So as a sort of a motivation for what types of systems we want to study with this method, one interesting candidate are photoreceptor proteins. In general, these mediate light responses in organisms, in living organisms. They tend to bind naturally occurring chromophores, such as flavin, you see here. Also things like biliverdin, which is in the case of the phytochrome, carotenoids, other things that can be built directly into the protein itself. They show sort of a reversible action in that they can go through many cycles. So they absorb light, undergo some sort of structural change, and then revert back to their dark state over time. And more recently, they've been exploited for optogenetic purposes, where they can be sort of engineered in sort of a plug and play fashion, where you can fuse together several different domains, where you have photosensing domains that sense the light, and then output domains that induce some particular biological function. This is, yeah. There are many examples of this type of optogenetic engineering. It's been successful in modulating protein-protein interactions, nuclear transport, neuron activity, gene expression, DNA binding, and many, many more. But the ultimate goal is sort of creating light-activated control of biochemical activity within living organisms. Sort of a special class of these photoreceptor proteins are the so-called love domains, which is sort of an abbreviation for light, oxygen, and voltage. So these are part of the phototropin photoreceptor in plants, and generally are sensitive to changes in either light intensity or lighting conditions or change in redox environment. So they allow the plant to be able to adapt to its environment. The LOVE2 domain in particular is an interesting one, which is shown here on the bottom right, where you have here a embedded flavin chromophore. This is flavin mononucleotide, and then a very large alpha helix, which is known as the J alpha helix, docked on top of this beta sheet opposite the chromophore. And then on the N-terminal side, you have a much smaller helix, which is known as the A prime alpha helix. This is from Avena sativa, which is actually oat. And so the flavin is a blue light absorbing chromophore. So upon absorption of blue light, you see this radical change in the absorption spectrum. And it was discovered through NMR that you get large, say, destabilization or unfolding of this J alpha helix in the lit state. And so this was a very attractive template for developing optogenetic constructs, given that most of the protein remains intact and you get this nice reversible structural change in the unfolding of the J alpha. So here's one example of how this has been exploited. So the group of Klaus Hahn, some time ago already in 2009, came out with the idea of fusing the RAC1 GPTase, which is involved in the modulation of actin and the sort of control of cytoskeletal dynamics in cells. So they fuse this to the J alpha helix. So in the dark state, RAC1 is held close to the LOVE protein itself. and unable to bind effectors in its local environment. However, in the light state, when it absorbs a blue photon, you see how it gets released and is able to make this binding. And the result is that you're able to sort of control cellular motion with light. So you see here on the left where the cell is migrating sort of towards the lower left side, and then the other side it's going the opposite direction, which is kind of a fascinating demonstration of how these photoreceptors can be used. But in general, from sort of the biophysical context, we want to ask the question: how do these sort of local dynamics near the chromophore following the photon absorption lead to these large-scale structural changes and action at a distance? So how does this signal get transduced through the protein and create such an effect? So a lot of work has been done on understanding this the photoactivation mechanism of ASLOVE2. And one particular feature that has been discovered is this formation of a cysteine adduct to the flavin itself, which occurs roughly in a couple of microseconds. This has been determined through vibrational spectroscopy, mainly, and other methods. So this is sort of thought to be the sort of the precursor, the trigger to the J alpha unfolding event. As I mentioned, in sort of in a former life, I was a spectroscopist, and I carried out some transient infrared measurements on this particular protein quite some time ago already now. And you can see how with vibrational spectroscopy, as with any spectroscopy, you get what appears to be quite a messy situation here. So you're looking at here decay-associated spectra from nanoseconds out to about a millisecond. And we resolve, say, several components. The black and the red are mainly due to the vibrations of the flavin itself. But as you see those decay, there are some interesting features that pop up. So first is this lower frequency band here, which is represents the formation of the cysteine adduct to the chromophore itself, and that occurs on the time scale of 10 microseconds. And then you see on the other side here, a hydrogen bond or signature of a hydrogen bond breaking, the small positive band here, which is the glutamine near the chromophore, which is on the previous slide. And finally, there's a large bleach or loss at around 1630 wave numbers, which we determined was actually had signed it to the J alpha band, the amide-1 band of the J alpha helix. And that occurs, or that loss occurs sort of in two steps, it appears. One coincident with the adduct formation, and then continuing in a few hundred microseconds. So we also compared sort of this infinite band, as I labeled here, to the steady state case, and this matches almost perfectly. So we're pretty confident that we capture most of the sort of the internal structural dynamics of the system. And we, as I mentioned, sort of so the adduct formation, the J alpha unfolding, possibly in two phases. We can sort of see the sequence of these events, but we don't get a lot of structural context. It's very difficult to model these data and pull out actual structures, which is what motivated us to turn to solution scattering, which is more of a direct probe of structure. So we had a beam time a few years back in the fall of 22 at the European XFEL at SPPSFX. And this is how our experiment was set up. We were working with 8 keV photons, and we used a nanosecond laser at 475 nanometers, which was orthogonal to the X-ray beam. And the AGIP detector had a distance of about 28 centimeters. The sample itself was delivered to the X-rays using a GDVN. This is one of the 3D printed GDVNs that are fairly common nowadays at the European Expo, commonly used. And due to the sort of the unique nature of this detector, which is able to read out at very high frequencies up to megahertz, in fact, we decided to employ kind of a unique illumination scheme where instead of doing like a traditional pump probe where we probe at a fixed delay many times, we decided to use the sort of strange bunch pulse structure of the European XFEL, where you have sort of bunches of up to, say, 300 pulses at megahertz repetition rate, separated by or running at 10 hertz. In this case, we were running at half a megahertz, so we get about 175 pulses. And our tactic was to pump alternating trains and then read out each individual pulse as a function of delay. So essentially, we pump sufficient sample to span this entire pulse train, and then read out each pulse, which gives us essentially 175 delays at one time, as opposed to doing it one at a time, which is the conventional approach. And so at 564 kilohertz, our separation between the pulses, our effective time resolution of the measurement would be about 1.77 microseconds. And sort of the goal, as I explained earlier, was to investigate the mechanism of this j-alpha helix unfolding. And some questions were, is it a multi-step process? Does the helix completely unfold? Does it simply just undock? And also, is there movement of this smaller N-terminal helix, the A prime alpha helix? So just to sort of re-emphasize how the illumination was done, because the nozzle is semi-transparent, we have to shoot, we have to illuminate the sample within the nozzle. So this is a 3D printed nozzle, as I mentioned, and the internal volume of the nozzle has to be large enough to essentially span this time interval. So in this case, it would be about 300 microseconds. So that translates to about 1 nanoliter of sample. And so our excitation spot was about 300 microns, overlapping with the bottom portion of the nozzle, and then our X-ray probe spot, which is just below the orifice. So you see here, then we have alternating light and dark trains, and these were subtracted directly from each other to yield the difference signal. So this was sort of like a one pump and multiple probe approach. This is the data itself, and these are sort of the time-gated scattering curves, scaled down at the lower angles because just to sort of emphasize the very large range of the measurement in terms of amplitude. And you see immediately that there's systematic evolution or clear evolution happening on all time scales, out to very wide angle, which was super interesting. And also the noise floor, looking at the pre-zero signal here, is very, very low, in roughly on the order of 1 to 10 to the minus 5, which is about an order of magnitude smaller than comparable measurements at a synchrotron. So that looked nice. Now we have to sort of think about how to understand the data, and the first thing we wanted to do was this sort of kinetic decomposition to figure out how many potential states might be involved. We first assumed just a simple two-state model of say A going to B, and found that didn't sufficiently capture the dynamics that were happening. So we added then a third state, and this sort of sequential fashion, which seemed to fit the data quite well. So state A here forms within the first probe pulse, so that's roughly 2 microseconds, and very close to the time scale of the adduct formation. And the final state, state C, which is clearly the dominant contribution, you see this is also scaled down as well here, the green curve, sort of culminates in about 300 microseconds. So by the end of the probe window. Whereas state B is sort of this, what I'm calling sort of a hidden intermediate, which hasn't been identified in previous spectroscopy measurements. And so due to the very low amplitude of the A and B states, it's very difficult to understand or to do any sort of modeling. So we focused mainly on the C state. We know that this timescale matches very well with the vibrational spectroscopy. So that was encouraging, but we really want to know, we were after some structural information. So, and this is sort of a difficult task due to the timescale. In many cases, you would do things like MD simulations, and unfortunately, that's not so easy for out to hundreds of microseconds to do pure MD simulations. You can do, of course, some types of non-equilibrium simulations or some tricks to try and force the system into this final state. But instead, we opted for a slightly different approach using AlphaFold. And in the beginning, the idea was to simply generate sort of oversample or aggressively sample and try and find structures with the unfolded helix. Turns out after, I don't know, like a few thousand candidate structures, we didn't find anything with the helix unfolded, which was just not so fun. So afterwards, the next step was to try and sort of force the helix to unfold, and we did so by inserting glycines to alternating residues in the helix. And using this sort of tactic, it seemed to destabilize the helix, and we were able to generate a pretty large data set of candidate structures. And then, so these among these structures, they were we calculated scattering curves, different scattering curves, using a dart structure. This was also generated from AlphaFold. And you see on the left, we sort of compare these candidate structures to the experimental data itself. There's one caveat here, because we don't know the absolute, or we don't know that there's sort of a scaling factor, right? So we had to do sort of a separate measurement to determine sort of a steady state SAX measurement with saturating illumination to determine the yield of our XFEL data. And that was done and determined to be roughly 15%. So what you see on the lower left here within this box are the sort of best fit structures, which entails, I think, about 6,000. And this is sort of like a plus or minus 5% window surrounding our 15% photoactivation yield. And then you see on the right how these line up with the experimental data, which is quite nice, I would say. On the bottom right, you have here the change in the radius gyration, which sort of peaks right around 6 angstroms, which seems reasonable. So we were quite happy with this. Let's say it's not the maybe the best approach, but we were able to gain some structural insight into into what was going on. So here is sort of a subset of those structures. I think like 20 structures or 30 structures on the right, and in all cases, you see that the both helices are unfolded, which is sort of what I expected. But also there were some small details that I found. In particular, in the dark state, the residues immediately preceding the helix sort of form this loop region here. Whereas in the light structures or the AlphaFold models, we see that this converts to more of a helical form. So potentially this has some sort of significance in terms of driving the reaction or driving the helix to unfold. That's that will require a little bit more work, I think, to fully understand how, in particular, this low amplitude intermediate state, how this gets translated into the helical domain from the beta sheet. So that's yet to be done. So in the end, this is the sort of reaction scheme that we came up with, where in around 2 microseconds, you get an attic formation, which we capture in the X-ray data, and then the helix unfolding occurs in roughly, say it terminates in 300 microseconds. This is difficult to fit because you have a large uncertainty in these time constants due to the small amplitude of the B state. So you say it occurs in a biphasic manner and culminates in roughly 300 microseconds, which is in strong agreement with the infrared data. So I wanted to just point out, a couple. It was how do we rationalize the difference in signal quality between the XFEL and a 4th generation synchrotron or a synchrotron source. And mainly this comes down to sort of the usable flux. So you see on top, if you were to take, in this case, we assume the Agra2 detector running at 5 kilohertz, giving you this sort of 200 microsecond integration time. On the microsecond time scale, you're unable to use all of the flux, right? So you have this idle flux in between the readout of the detector. Whereas in the case of the XFEL, we can read each individual pulse, and each pulse contains about a few orders of magnitude more photons. So this is how we sort of rationalize the lower noise floor that we're seeing in this experiment compared to analogous experiments at synchrotrons. On the bottom, there's this plot where we compare sort of integration time versus sort of the number of available X-ray photons, and you see at microsecond time scales, the XFEL increases and then plateaus because of this strange pulse structure, this bunch pulse structure at 10 hertz, whereas the synchrotron slowly catches up. So on second time scales, the number of photons is roughly equivalent between the two light sources, but on short time scales, the XFEL is higher. Now this is going to change hopefully fairly soon when we have the opening of LCLS2, which will, instead of running at 10 hertz, will run at continuously with megahertz X-ray pulses, and in that case will provide even more potential for expanding or for increasing the sensitivity of transient solution scattering measurements. So just to sort of summarize, we investigated the LOVE2 or AS-LOVE2 J alpha helix unfolding mechanism with transient wax by applying an XFEL probe source. In this way, we could achieve dense time separating in the microsecond to millisecond range by exploiting this bunch pulse structure, as I mentioned. And this type of illumination scheme, or this sort of one pump mini probe illumination scheme, might be extended to other types of scattering methods such as SFX, crystallography, or crystallography, since it requires almost no modification to the setup other than just changing the position of the illumination light. So it's fairly easy to implement. And microsecond timescales are quite important for the study of, in particular, photoreceptor proteins, where lots of structural changes are happening. So I expect that it might be useful for other methods. So the enhanced sensitivity of the low noise floor here might enable measurements on dilute solutions, or possibly out to higher, wider angles or extended Q range, which can give us slightly higher resolution, maybe picture structural models. Of course, it comes at a trade-off, being that the sample consumption is fairly high. I think in our experiment, we used about, the data took about 3.5 hours to collect. And because the GDVNs run, you're not able to recirculate the sample. At least in Hamburg, they don't have this set up. I want to say like 70 milligrams of protein we used for the entire measurement. Also, XFEL beam time is competitive. It's very difficult to make these measurements, and comparatively probably more expensive than doing it at a synchrotron. So I guess I'm not saying it's a one size fits all approach, but in certain situations it might make sense to employ uh, uh, to do this type of transient wax or SAX at an XFEL. So with that, these are the people that did the work. We collaborate closely with Sebastian Wessenhoff, and Leonardo was one of the students who did the AlphaFold modeling. And also, of course, want to thank the the XFEL staff and all the participants of the beam time, which were there too many to mention, unfortunately, and the funding agencies. And also thank you for your attention.